What is it all about? Well, believe it or not, genetic engineering started approximately 35 years ago in a delicatessen over a pastrami sandwich, and that is the truth. It started at a meeting in Hawaii where two individuals came together, Stanley Cohen and Herbert Boyer. Stanley Cohen was a professor at Stanford University. Herb Boyer was a professor at University of California, San Francisco. And they were both working very independently on totally different things. And this is what's remarkable about science. Sometimes people are working in this house. Sometimes people are working in another house. They come together, and when they come together, they merge their ideas and invent something that had never been thought about before. At this fateful meeting in Hawaii, Stanley Cohen and Herbert Boyer came together, and it was a eureka moment in which genetic engineering was invented. Prior to this moment, which was in 1973, genetic engineering wasn't even thought of by anybody. Up until that time, no one could isolate a specific gene. You'll see what I mean about that in a second, because all of your genes are next to each other in chromosomes. So prior to that particular time, no one could isolate a specific gene. What Stanley Cohen and Herbert Boyer were working on were two very important problems. Herbert Boyer was working on enzymes. Enzymes are proteins that catalyze enzymatic reactions. Enzymes that are able to chop DNA into pieces. And furthermore, is able to chop DNA into pieces because there were enzymes that were in bacterial cells that used those enzymes to protect themselves against viral invasion. And so these enzymes are a primitive form of defense mechanism that allowed the cells that they're in to protect themselves from the invasion of viruses. On the other hand, Stanley Cohen was working on an extremely important problem. The problem of, in fact, antibiotic resistance. Now, all of you know that antibiotics are being used and are used to kill off bacterial diseases. And so he was trying to find out why bacteria acquire resistance to antibiotics. And antibiotic resistance remains one of the most difficult public health challenges even today. Because for many diseases, antibiotics that we have don't work. And that's, like a really serious thing. Like MRSA, multi-drug resistant staph, it's serious. Because if you get that thing, God forbid, there's almost nothing that's going to be able to contain it. So Stanley Cohen was working on antibiotic resistance. Herbert Boyer was working on these weird enzymes that people didn't really think had any importance. They came together, and when they came together, what they did was they had a novel idea. And let me tell you what that novel idea was. Inside these bacteria, and we're going to go over this in living detail, this is just an introduction. Inside these bacteria, which are single cells, there is a small little thing called a plasmid. It's a small extra piece of DNA which is not part of the chromosome of the bacteria but replicates independently as these little red circles show. It just so happens that those extra pieces of DNA contain genes. Not very many of them, but they contain genes. And it just so happens that those extra pieces of DNA just so happen to have the genes that specify antibiotic resistance. And one of the reasons why antibiotic resistance can move through bacterial populations is that these little circles, as you can see, when the bacteria mate, here's a bacteria that don't have those circles. Here's a bacteria that have those circles. These circles can move from this bacteria into that bacteria. And if they carry a gene for antibiotic resistance, this cell, which was originally antibiotic resistance, gives rise to another cell, which is now also antibiotic resistance. And these things can spread. And they spread in the water. They spread in hospital sewages that come out of hospitals. They spread everywhere. And the reason why you can spread this antibiotic resistance is because they're on these little extra chromosomal circles called plasmids. And that's what Stanley Cohen was working with. And he was trying to understand what those plasmids were, what those genes were, and how those genes allowed those particular bacteria to be resistant against an antibiotic which will kill the bacteria. That's what the antibiotic does. The genes on these plasmids detoxify those antibiotics and essentially allow the bacteria to survive. On the other hand, Herbert Boyer was studying these killer viruses. These are viruses that infect bacteria. These viruses inject their DNA into the bacteria, and the DNA then takes over the bacterial cells, replicates itself, and eventually kills the bacteria. Now imagine if the bacteria didn't have a defense mechanism against that, you wouldn't have any bacteria. In other words, the virus would infect the bacteria, kill all the bacteria, the virus wouldn't be able to replicate anymore, and there wouldn't be any more bacteria. So there is a play between the virus trying to infect the bacteria and the bacteria defending itself against the virus. And the way in which the bacteria defended itself against the virus was these things called restriction enzymes, these little green balls. And these are little proteins in the bacterial cell which has the ability to chop up the DNA of the virus, as you can see, into pieces. If the DNA of the virus is chopped up into pieces, it can't do anything. It's essentially dead. And so these little enzymes that Herb Boyer was studying were a defense mechanism to prevent the virus from killing the bacteria. Now you can ask the question of how come these little enzymes don't kill the genes in the bacteria per se? And the answer is, in short, is that the genes in the bacteria have these little purple marks on them that I'm not going to talk very much about, but those purple marks protect the DNA of the bacteria from these little green enzymes. Notice that the DNA of the virus doesn't have those little purple markers, and therefore they're exposed, they're naked, and these enzymes can chop the DNA into pieces and kill the virus. So Herb Boyer was studying these particular enzymes. Stanley Cohn was studying these little plasmids and studying the mechanism of how these plasmids work. They came together in this delicatessen over a pastrami sandwich in Hawaii, and they said, hey, we think that these enzymes called restriction enzymes can recognize any DNA, not just viral DNA, can recognize any DNA. And what was the key was that these enzymes didn't recognize just any part of the DNA. These enzymes recognize a specific chemical composition in the DNA. It recognized, and you'll see what we mean by this later on this week. These enzymes weren't just non-specific and chopped up the DNA anywhere. In fact, these enzymes bound to the DNA and chopped the DNA into very specific pieces. And so they reasoned that if these enzymes could chop the viral DNA into different pieces, these enzymes would have the ability to chop any DNA into pieces. But more than that, they had the ability to chop the DNA into very specific points that I'll call A right here for simplicity. And any particular DNA that has this location A could be recognized by this enzyme. And if that was the case, as you will see, when the enzyme chops the DNA into these pieces, notice here's DNA 1 and here's DNA 2, this enzyme can chop the DNA into this position. This enzyme can chop the DNA into the same position. And when it does that, as you will see this week, in fact, tomorrow in discussion, and this is going to be a piece of cake for you guys, what it allows is that the DNA, the blue DNA, can be joined with the yellow DNA to form a hybrid molecule. Now, I know that seems very esoteric. I know it seems very complicated. I know it seems very Byzantine. I know it seems rather oblique. I know to many of you that are political science and communication studies majors, that sounds really complicated, but in fact it isn't. The epiphany was that they recognized that these enzymes could recognize a particular place in the DNA of any creature, chop it in a particular place, and then you could take those two very different DNAs and join them together in that same place to make a hybrid DNA molecule. Where did the plasmid come in? The plasmid came in because if you did this with a plasmid, what that would mean is that these plasmids behave as their own little chromosomes. And what would happen is if you did this with a little plasmid, as you will see again this week, you could take one plasmid. Here it is. You can take the blue plasmid, let's say that has a particular kind of antibiotic resistant gene. You can take the red plasmid, which has a different antibiotic resistance gene. You can open those plasmids in the exact same place with this enzyme. And then you can recombine those plasmids because they'll join in the place in which they were cut by the enzyme. And now you have a hybrid green-red plasmid. But notice instead of having one antibiotic resistance gene, it now has two antibiotic resistance genes. And furthermore, what they decided and what they determined was that you could take this, do it as naked DNA in a test tube, and then reinsert the DNA back into the cell. Let me say that again. They take the bacterial cell. They isolate, like we did, the plasmids from bacteria cell A and bacterial cell B. They take the same enzyme. They chop the plasmid A with this enzyme, chop the plasmid B with this enzyme, and in a test tube, they join them together. The DNAs of these two different plasmids join. And then they take this and are able to put it back inside a cell. And instead of having two different bacteria, each of which is resistant to one antibiotic resistant gene, we now have a bacteria that's resistant to two antibiotic resistant genes. And in fact, we've made a new organism, in a sense, because we're able to combine the DNA of, let's say, two different kinds of bacteria which have two different kinds of antibiotic resistant genes and able to put them in the cell of one organism. Now, you're going to learn more about that life in tomorrow than you're ever going to want to know for the rest of your life. So hold your question until after you read the articles for discussion, and then we'll talk a little bit more about it as you go. By the time this quarter is over, this is going to be like reading Run, Dick, Jane to you. That's how easy it's going to be. And I know you don't see that now, but trust me, that's going to be the case. So that is the origin of genetic engineering. So why would you want to ever do anything like this? I mean, what is the whole point of doing anything like this? Well, your genes, both here at UCLA and at UC Davis, your genes are on chromosomes. And your genes are strung together in long strands of DNA. And so on that long strand of DNA, you have gene A, you have gene B, you have gene C, you have gene D, you have gene E, you have all of these strands of DNA. Kelly, could you do me a big favor and erase that board for me? Okay? And so I can use it. And so the point here is prior to the invention of this technology, one could never isolate a gene. One could never study a gene in a test tube. You could study all of the genes, like we just did, as a collection, but you could never look at a gene, you could never a particular gene, you can never study it, pick it apart, or manipulate it prior to this particular invention. And so the revolution of Cohn and Boyer in 1973 allowed being able to take a piece of human DNA, digest it with these magical enzymes, get a specific piece of DNA, but more than that, a particular gene. And let's say in this case the gene specifying insulin. And then stick it in these plasmids, which again you will learn can replicate by themselves in a bacterial cell. And now we have a human gene isolated by itself. On this plasmid, there's no other human genes. Now we can study its structure. We can study what protein it specifies, because genes specify proteins. We can stick it in a bacterial cell and have the bacterial cell make tons of this particular protein, in this case insulin, and now we can use this bacteria and this insulin gene in order to make a drug, insulin, and be able to treat human beings with that have diabetes. And so the purpose of this genetic engineering revolution was to be able, for the first time, isolate particular genes, grow them in infinite quantities in bacteria, and be able to study them and understand what their structures are. If you open any biology book, how many of you have taken biology not so recently, but within the last few years? You've had a book. In that book, probably 80% of knowledge came from using genetic engineering in one way or another. So it created a revolution in being able to manipulate genes, and the technology has evolved so far to the point where we can do anything we want in terms of manipulation. Any gene, as I said, can be put in any organism, can be rearranged, can be mixed, can be matched. We can do anything with it, and that's really where it's at. Now let me show you some. Leifin, how you doing? You want to take a break? Or you want me to keep going? How many people want to take a break? Good, I'm going to keep going. So let me show you some examples of what I'm talking about. No. So what can we do with genetic engineering? Dora, do you have any questions?
No.
Picking on you. Any questions up at UC Davis? I love this interaction. Ryan, do you have any questions? You know all about this technology?
A little.
It's such a shame I wasn't on your PhD oral exam. You use it all the time, don't you?
Oh, every day.
Yeah, I know, exactly. Here. So what can we do with genetic engineering? And let me give you some startling examples. So the point here is that we have a whole bunch of genes on chromosomes. So here's a map of the X chromosome of humans. Here are different genes. Notice that they're strung together in a linear order. Nicholas, why do you think those genes are strung together in a linear order? Do you think that chromosome has one piece of DNA or many pieces of DNA? For $1 million tax-free in your checking account tomorrow, be able to use, what's the answer? One piece or many pieces? What do you think the simplest thing is? Simplest.
One piece?
Exactly. There's one piece of DNA, you just won $1 million per one chromosome, all strung together in that little thing that you saw. Of course, when you had that sperm DNA, it was 22 chromosomes plus a Y chromosome, so you had, 23 strings independent. But the point here is that the DNA is strung along from top to bottom. Well, with genetic engineering, you can now isolate this gene, this gene, this gene, this gene, this gene. How do you think we know what those genes are? We know what those genes are and can make this figure because someone, in some point over the last 35 years, isolated these genes by figure because someone, in some point over the last 35 years, isolated these genes by. The point here is why do you want to do something like this? And here's the crux of it. The crux of this, Thomas, is here's the key. In order to get enough gene to study, and in order to get genes away from other genes, you have to use genetic engineering. So if you have all of these genes strung along together on the X chromosome, and you want to get a specific gene, you need the restriction enzymes, and you need this plasmid, so to speak. Because then you can tease them away from all the other genes, and now you can put them in a bacteria, but because the bacteria replicate, you can make infinite quantities of that gene. There's only a tiny amount of each gene in every cell. We can study it, but you can't get a lot of it. And so genetic engineering allows you to isolate a particular gene, and as you can see on the screen and on your handouts, be able to make tons of it, and being able to study the gene in infinite quantities, be able to have as much of it you want, to be able to do whatever you want, to be able to make a drug out of it, to be able to study the structure of the gene, be able to study the chemical composition of the gene in order to use it to diagnose a specific disease, use it to identify a particular individual. You could use it to correct a disease, use it to make new crops. You can do all kinds of things that you will learn about in this class. Seems very abstract now. And furthermore, as I said, and I'm not going to go into this in depth, this technology has changed our lives enormously because it allowed new technologies. For example, drugs, growth hormone. Humans treat people who have growth disorders. Insulin, treat people who have diabetes. Diagnose cancer early. Treat cancer early. Diagnose other kinds of diseases. Sequence the human genome. Sequence the genome of all different kinds of creatures. It's led to all kinds of absolutely amazing applications. Started a biotechnology industry, started DNA testing in the law, allowed getting ancient DNA from the woolly mammoth and the Neanderthals, allowed the beginnings of synthetic biology. Everything that we do in this day and age traces back and has its origins in this amazing revolution called genetic engineering. In 1972, just before I came to UCLA, we used to do experiments on whole genomes. We didn't know what a gene was. When this technology was invented in 1973, soon thereafter we were able to isolate genes and understand what they were and what they did. That was a huge revolution in terms of understanding what genes are made of, what genomes are made of, and what cells do. But more than that, as you can see, this technology has led to profound social, legal, cultural changes. As I showed you previously, perfume named DNA. DNA is part of the culture. You hear a really good sports hero who can do something really well. They say, it's in her or his DNA. It's part of our culture. But more than that, what you can see on this particular slide is that in many ways we have patenting genes in living organisms. I can own your gene. I can take one of your genes and patent it. What do you think about that? And we're going to talk about that. So it led to patenting genes. It led to genetically modified organisms, plants, crops, supercrops that, as I'll show you in a few seconds, have genes in them that they weren't born with. It led to labeling issues of genetically modified foods. It led to regulating the release of genetically modified organisms into the environment. It led to legal issues of genetic testing, databases. It leads to laws in Congress which have been passed preventing genetic discrimination. It's led to the ability to do gender selection. It led to gene therapy, as you'll see in a second, curing diseases. It led to all of these very profound issues, including synthetic genomes and constructing new organisms, all had its apical point of invention in 1973. Let me show you some startling examples leading up to humans. And this is, what I'm going to show you is going to seem very startling to you guys. I'm kind of laughing here. Because John and I know that what I'm going to show you is child's play. Well, I'm going to show you child's play stuff. It's going to seem highly scientifically incredible to you guys, but it's child's play compared to what we can do now. And that's because this technology has advanced in an enormous way. So let me start with some baby stuff. We have a jellyfish that has a protein. Proteins carry out the work of the cells. Proteins carry out the work of the genes. We have this jellyfish, and it exists in the ocean, and it has a protein that when light shines on the protein, it fluoresces. Green, red, orange, all kinds of different colors. Well, that protein is encoded by one of the genes in the jellyfish genome. In fact, the Nobel Prize was just given for the person that didn't isolate the jellyfish protein that fluoresced, but had the idea of using it as a tool for being able to study living processes and be able to visualize them. That Nobel Prize was just given just recently. So the point here is we have this fluorescent protein. We can use genetic engineering to get the gene for that fluorescent protein out of the jellyfish. We use those restriction enzymes. We cut the DNA. We use those little plasmids, those little red circles. We insert the jellyfish fluorescent protein into those little circles. And we now have, let's say, a bacteria that has this jellyfish gene in it, and we can do anything we want with it. But, you know, why stick it in a bacteria? How about taking this jellyfish gene and sticking it in a real fish? So there is the egg of a fish. It happens to be a zebrafish. Let's say we take the jellyfish gene and we take a needle and we put that particular needle next to that egg and we inject it. So we're taking a naked piece of DNA and we're now injecting it in this particular egg. And that gene goes into the egg and is taken up by the egg, and then hopefully this is going to work, but maybe it won't. There it goes. And notice that the embryo is developing. That is a fish embryo that was developing. And notice that fish embryo that's developing, if I can get that thing going again, fish embryo which was developing, notice that in one part, there it is, it's fluorescing green. So what we've done is we've taken a gene from a jellyfish, which is 500 million years divergent from a real fish. We've taken that gene out by genetic engineering and stuck it in a real fish, and eventually you can get back a fish that glows. And in fact, you can get a fish that glows. These are called the glowfish. You can't buy them in fish stores in California because California has a law banning the sale of any genetically engineered fish. And we're going to talk about why in this class. But you could buy them in Arizona, you could buy them in Ohio, you can buy them in Nevada, but you can't buy them here. But that's clearly a genetically engineered fish. It glows green. But more than that, you can get fish that'll grow red, green, yellow, and all kinds of different colors. But the point here is Marvin likes to use email a lot. The point here is Marvin, you relaxed, Marvin? You sure? You're pretty tense the last couple days. What'd you send me, about 50 emails? Did you? You're calmed down now? Good man. The point here is that this fish now has a gene in it that it wasn't born with. That's genetic engineering. And that gene works. The jellyfish gene works in the real fish. Put a light on it, it glows green. Well, that's baby stuff. You get all kinds of different fish of all kinds of different color. Orange, yellow, green. Wonderful. You can make a whole aquarium of genetically engineered little fish just by what kind of fluorescent jellyfish gene you stick in the fish. But, you know, let's move up on the scale of things. And let's say, well, I don't want to put it in a fish. I want to put this thing in a fruit fly. And so now you can see we have a fruit fly, these insects, in which we have the jellyfish gene engineered into them. And notice when you shine the light on them, they glow. So now not only can we take the jellyfish gene out of the jellyfish and stick it in a real fish, we can stick it in an insect, and the insect glows. It's like a firefly, except it's a glow fly. It's a genetically engineered fruit fly. Well, that's okay. How about mice? Now we're getting close to humans. Notice that these mice have a gene in them in which they were not born with. When these mice are born, when these mice were developing, they had a jellyfish gene in them. And that jellyfish gene is active all throughout the mouse. And what you can see is that the light can be shined on those things, and notice that you see green tails, green noses. Juliana, microphone. Can you make what? Can you make the fur glow? Probably not, because the fur is kind of opaque, and the fur really isn't a cell. The cell of the fur is at the base, and the fur is really just protein. You have to have this in a cell so that the protein can be in the cell, and then when you shine the light on the cells, the light goes green. So probably not fur. We can do other things with fur, but not that. Albert, my man, feel free to ask some questions up at Davis.
What happens if, like, this...
Turn the mic up, please.
What happens if the glow mouse mates with a normal mouse?
Well, what do you think? What's your hypothesis, Albert? That's a very good question. I'm not going to answer it. I want you to answer it. And if you can't answer it, call on someone in the class to answer it.
Doesn't matter if, like...
Well, answer your question.
Could the gene be dominant in some... Like,
well, you're answering your question with a question, so I want you to answer the first question. If you took the mouse that has the glowfish gene and you mated it with a mouse that didn't, you think the gene would go away?
No.
What would happen to the gene?
It would go into the new mouse.
Yeah, it would go into the new mice. It would be passed on from generation to generation. Depending upon whether there's one copy of it or two copies, it's inherited because it's part of the chromosomes of the mouse. It's an excellent question. Okay, well, we have it in the mice. Well, big deal. How about if we stick it in a monkey? That's a rhesus monkey. That's one of the closest relatives to human beings. We have a red fluorescent protein engineered into the chromosomes of a rhesus monkey. And in fact, it's not a very good picture, but that pink is actually red fluorescent protein. The point is that we can make a fluorescent human if we engineered a human being, but we don't do that because there's many ethical, moral, and other kinds of issues that we have to deal with. Now we can also put it in a plant. So you can make a glow plant. The point here is that one gene from the jellyfish can be taken from the jellyfish into the fruit fly, into the regular fish, into a plant, into a mammal, a mouse, into another mammal, a monkey. You can do anything with it. That's the point of genetic engineering. The point here is you can isolate a gene from all the other genes in the jellyfish. You can make gobs of it, and you can combine it with the DNA of other organisms, and I love that name, Garden of Eden, and you can get it to work. That's really something remarkable. And these are very simple baby examples. And I'm going to show you some other ones that are a little bit more complex, but still pretty simple. Okay, what about plants? All of you have heard about genetically modified organisms. All of you heard about plants that have been engineered for new genes. We're going to talk a lot about that in this class, and I'm not going to go into the technology. I'm not going to go into the reasons why and how and the pros and the cons. I just want to show you that you can take a gene from bacteria and you can stick it in a plant and you can make it absolutely resistant to insects. In other words, you're never going to have to spray that plant with insecticides to get rid of the insects because it's going to have a built-in, so to speak, insecticide. So what you're seeing on the screen is a little poster that says protect against cabbage crops. And if you have little worms on your cabbage crop in your gardens, you go to the store and you get little spores from a bacteria called Bacillus thuringiensis, and those little spores go on the leaves of the plant, and when the, let's say, insects come along, they eat those spores and they get a massive case of indigestion. Now, this bacteria has genes in it that make a protein that have the ability to kill caterpillars, harmless to human beings. It's been used by organic farmers for the last 100 years and is still being used. Now, what happens if we isolate that gene using genetic engineering? We isolate that gene from this bacteria, isolate it from all the other genes, and put it in the plant. And we get it, in fact, to grow in all different parts of the plant. The hypothesis is that if it works, that protein is now going to be produced by the plant. We now have a bacterial protein being produced by a plant. And you'll find out why in this class. And therefore, when an insect bites on the plant, they get a massive case of indigestion. Now, why would you want to do anything like this? Well, if you ever have been in the developing world, you will know that although we only spend 15% of our income on food, in many parts of Africa and Asia and Latin America, they spend 85% of their income on food. And if you're a farmer in one of these poor parts of the world and you have an insect infestation, let's say locusts, like in the Bible or anyplace else, it's going to wipe out your crop. And if it wipes out your crop, not only are you going to lose your income, but you're going to lose your food supply. And furthermore, in order to get rid of those insects, you're going to have to use insecticides. And I can guarantee you it ain't the guys that have these poisonous insecticides on their back. It's the women and children who are in the field with poisonous chemicals spraying all of these plants to get rid of these insects in order to be able to survive. But if you could use genetic engineering to engineer those plants so that you wouldn't have to put insecticides on them, that means that women wouldn't have to work in the field and could do other things and would free them to do a lot of other stuff. It means that the poor farmers don't have to buy insecticides, so that means their incomes will go up. It means that they don't have to spray their fields with all kinds of noxious stuff. And furthermore, the food that they get out is absolutely chemically free. If you could do all of that, you'd do an awful lot for farming in the developing world and in the developed world. And in fact, that's been done and was done many, many, many years ago, as you will see, I hope, in this clip, which I should have practiced before I got to the class, because it looks like, there we go. Watch. Genetically engineered plant on your right. Non-genetically engineered plant on your left. Worm was put on both plants. Notice what's happening to the plant on the left. It's getting destroyed. Notice what's happening on the plant on the right. Nothing. Imagine if you were in the field someplace in the developing world and your crop was being eaten by those insects. You're going to lose your entire crop, your entire income, and your entire food source. The only way to get rid of those before this technology was to spray. Notice how fat that worm is from eating those leaves. Notice here there's a couple holes and a little tiny dead larva right there. Massive case of indigestion. The point here is that using this technology can be able to make insect-resistant plants and be able to make a crop which is essentially insect-free. We now have a bacterial gene in a plant allowing it to be free of insects. And in fact, this has now been commercialized. And in fact, most of the corn that you eat in potato chips, most of the corn that you get in the store, generally will have been engineered with this particular protein and is insect-free. Yes, my man. I need a microphone.
Isn't it also true that they've genetically engineered these plants so that they only last one crop season?
No, that's not true. We'll get into that in a couple of weeks. Yeah, that's absolutely not true. That's a little myth that you hear on the internet someplace. Go ahead.
What's the implication?
Rose.
Yes. What's the implication of this with, like, say that the insects don't survive? Is there any, like, ecological issue with that, like predators and et cetera?
Yeah, we're going to talk all about that. Imagine that you're out there in a field spraying with insecticides. Think a lot of insects get killed with those insecticides? Yeah. An awful lot. This protein only targets one insect. It's highly specific. And so the consequences are what one would worry about, and we're going to talk an awful lot about this in 2 weeks, is insect resistance. But you also get insect resistance to insecticides. And so it's kind of like antibiotic resistance. You have to mitigate the benefits with, let's say the harm. In this case, there's very little harm because in order to have these things released into the field, you have to have a lot of testing. But more than that, you have to mitigate against resistance. And so what the farmer does is he plants his genetically engineered insect-resistant corn, and around the outside of his field of genetically engineered insect-resistant corn, he plots a refuge of a few acres of non-genetically engineered corn. And that serves as a refuge for insects to be able to feed and be able to survive in the wild without being killed by either insecticide, which is no longer used, or by this particular protein. But we're going to get into that, and we're going to get into all of these different issues. Is it Stephen or Steve? We're going to get into those issues soon. So what else can be done with genetic engineering? Well, we can make drugs not only in bacteria, but we can make drugs in mammals, like in goats, sheep. And that's becoming a big industry, and you're going to learn about that this quarter. But more than that, what's really important for you to understand is that. How you doing up in UC Davis? Are you bored, Dora?
No.
Not at all?
No.
Maybe you'll get a chance to come down and visit us during the course of this quarter. Wouldn't that be fun? And sit in this class. Same with you, Faria. We'll see what we can do about that. Maybe we'll get a couple UCLA kids to go up there and see how it is on the other end. How many of you have been up to Davis? It's like going in the middle of a cornfield, isn't it? In any case, sorry,
Tomato
John.
field.
Huh?
Tomato field.
Yeah, tomato field, exactly. Okay, here's the point. Catherine, what would you say if I told you that there were human beings walking around your age that have genes in them that they weren't born with? Got to turn the mics up a bit. Yeah.
Good for them.
So, would you think that it was pretty remarkable that we had genetically engineered human beings?
Yes.
Dora, what do you think about genetically engineered human beings? Does that scare you in any way?
A little bit.
Why?
eugenics and stuff.
Uh-huh.
Like thinking about how people can have like a superhuman or like preferred.
Do you think that
traits
we
of-
do anything like eugenics? Do you know anyone who dyes their hair to make themselves a little bit, younger or whatever?
Yes.
Do you know anyone who has plastic surgery to make themselves, have, different facial features or whatever?
Yeah.
Do you know anyone who tries to get their kids into the best schools like UCLA and UC Davis and others?
Yes.
Do you know anyone who any parents that try and get their kids into private schools and the best public schools possible?
Yeah.
Do you know any parents that want to have, the brightest and the best and the richest and the most successful kids on the face of this earth?
Yeah.
What would you say if they could choose, not by, let's say, genetic engineering, but if they did, kids that might be a little brighter, might be a little, I won't say prettier or whatever, because that's, a value judgment, but let's say a little bit brighter, and let's say that there was no harmful effects of this technology, would you use it for your kids if it gave them an edge over all the other kids in getting into some fantastic place and they had a sure shot for getting into medical school and they'd be able to make $150,000 a year when they graduated, have a wonderful life, and there'd be no harm and no ill effects? Would you think about it or not?
Probably.
Yeah, exactly. We all would. The point here is that when you think about eugenics, you think about all these monsters and you think about all these negative effects, but you don't think about all the things that we do to enhance ourselves, not using genetics in a very normal way, which is totally accepted, like using growth hormone and other kinds of things in order to give people, an advantage. So the point here is that what happens if, God forbid, Dora, and I mean that sincerely and not in any sarcastic way, someone that you know gave birth to a child that only had 1 year to live. And let's say that child was born with an incurable genetic disease. And it was an incurable genetic disease due to a defect in a gene which prevented that child from having an immune system. And let's say by genetic engineering you had the normal gene in your hand. And you knew if you stuck that gene in that child that will die in 12 months, it would be able to have a normal life, but you're having to genetically engineer them. Would you want to do that or not?
I probably would.
Yeah, well, there are kids walking around today, a lot of them, and here's one of them right here. This individual has a gene in it that she was not born with. She was born with a disease called severe combined immunodeficiency, which is a defect in a gene called ADA, adenosine deaminase, which happens to produce a very severe defect in your immune system. And prior to the ability of using this technology, these little babies had to live in a bubble. They couldn't be touched by their parents or anybody else. They had no immune system, and if they got any infection, they died. And generally, longevity of these children was 2 years. With the advent of genetic engineering in human beings, and this is, of course, one of the only known notable cases until recently, that individual is cured. So we not only have genetically engineered plants, we not only have genetically engineered fruit flies, we not only have genetically engineered fish, we not only have genetically engineered bacteria, we not only have genetically engineered monkeys, we have genetically engineered human beings, meaning we can genetically engineer anything and anyone using this technology. Now, remember Eden. We talked about the Y chromosome and the X chromosome. Now, what happens if, well, why do you think a male is a male? What's your hypothesis?
Because the Y chromosome produces, it has encryptions for proteins that make it, and hormones that make it different from a female.
Okay, so that's very good. So the Y chromosome has a certain composition of genes that kicks off a process that turns the developing human into a male. And in fact, what you're saying is that the ground state is a female, and that's true. Which brings me to the Bible in a second. If the ground state was a female, well, I'll get to that in a second. The point here is that we have all of these 22 chromosomes that are called autosomes in your in your nuclei. We have an X chromosome and a Y chromosome. And females have 2 copies of the X chromosome, and males have 1 X and 1 Y. And on the Y is a gene called sex-determining region Y gene, SRY. And that gene has been isolated by genetic engineering. Now, what would you say, Eden, if I gave you that gene and I stuck that gene into a cellular XX female cell? I took that gene, I put it in an egg that didn't have a Y chromosome. What do you think might happen? Let's say it's in a mouse. What's your hypothesis?
Well, aren't there, I don't know, isn't
Just
that
give me a
genetic-
simple hypothesis.
They would have male and female organs?
Well, not necessarily. You just told me that the Y chromosome produced a series of events
Right.
that led to the production of a male.
Well,
So what would happen if you put this one gene into the cell of this XX mouse, which was destined to be a female, but now you presented it with this Y gene, male? What would you think?
I guess it would, or I think it would have, it would have male traits. Or it
What
have
would be the male trait that it
or-
would
or
have?
Testosterone.
What would be the male trait that it would have? Yes, it has a hormone testosterone, but so do you. You have testosterone as well.
Male organs.
Yeah, that's exactly right, called a testis and a penis. And so they would have male organ. So now let's say that we take this gene from a human, not a mouse, but we stick it in a mouse that's destined to become a female, and notice that chromosomally we have an XX female, but I think that that's a pretty good penis right there. And so we now have transformed a genetically female individual into a male. Yes?
Isn't that like Jamie Lee Curtis?
Pardon?
Isn't that like Jamie Lee Curtis?
Jamie Lee Curtis is a little bit different. Jamie Lee Curtis has a Y chromosome, okay? But what would be defective on her Y chromosome? What would be the mutation in Jamie Lee Curtis's Y chromosome?
The expression of male organs.
No, come back to what we're talking about. We're not talking about organs. What's the topic that we're talking about? Starts with a G. So what gene is defective in Jamie Lee Curtis? She has a Y chromosome. Well, what's it called? We just talked about it. It's on your handout. What's it called? Male SRY gene. So Jamie Lee Curtis is chromosomally XY, but is a female because that gene has either been deleted or that gene has been mutated. And in fact, up until recently, female athletes in the Olympics were eliminated if they had XY chromosomes, when in fact they were completely female because their SRY gene was defective. And so the point here is that one can get this gene and engineer this gene and do all kinds of interesting things with it. Samantha, what a nice name. Turn it on. No, it's not on. Yeah, but it doesn't matter. You got to push the button.
Okay, got it. So you're saying you have an XX female and
I'm not saying
okay,
anything.
you showed the 2 XX females.
I haven't shown anything. You tell me.
Okay, so you have 2 XX females and you add the Y. Are you adding the Y chromosome or
What
are you
am I
adding
adding?
just the SRY gene?
SRY gene.
All right, so the sex determining region gene, if you're sending, so it's not the same as like Klinefelter's. It's not the same as Klinefelter's when you have it in a
Let's
human.
not even bring it up. That's a whole different ballgame.
Okay, that's a different thing.
Totally different.
Okay, so pretty much I was just confused because like on the SRY gene, it decodes for certain like hormones, and it stops, unlike the SRY gene, it stops the production. Like in a woman, there's Müllerian, like there's the Müllerian, and then the man, there's the Wolffian ducts, and like that's the difference between a man and a woman. And you're born with both, and then whether you're a male or a female.
Let's cut to the chase.
What I'm asking is like, if you add that in there, aren't you still gonna have? How are you gonna have both?
Yeah.
Can you have both parts?
Yeah. The point here is that it's a good question. Let's not confuse it with all the things that you've learned about all these physiological stuff. There's one gene. There's one gene. It's called the SRY gene we're going to learn about. That gene dictates the entire male developmental pathway. and it overrides every single thing that would be considered to be female. And so that gene makes a protein, and that protein sets off a cascade of events which allows that embryo to become a male. And all the other female parts never even develop. Never even develop. And so it has nothing to do with this, that, and whatever. Go ahead, Juliana.
Is that gene identical in mice and humans?
That gene, no. Fruit flies don't have an SRY gene because they have a sex-determining mechanism that's slightly different. But it's the same in all mammals, so to speak. Horses and goats and sheep and mice, it's basically the same because it originated way, 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 way back in evolution. Okay, so we can take a male mouse and take a female mouse and turn it into a male. And so I just thought, I just put this up. We'd say, well, the ground state of mammalian development is a female. And it takes one gene to switch from a female to a male, in a sense. One gene switches I have that wrong. One gene switches development from a female to a male. It's very simple. And furthermore, if you go back and you look at this quote from the Bible, it says that Adam was created by God, and then Eve was created from the rib of Adam. So the question is, where's Eve's Y chromosome? What happened to Eve's Y chromosome? And that's something you should think about, because the rib of Adam would have a Y chromosome. And if Eve was created from Adam, then there had to be either a Y chromosome or the SRY gene had to have been lost in some way. And so that's something that you should ponder from a philosophical point of view. Now let's look at some really old technology. What about making Mighty Mouse? So I've given you an example of the glowfish. I've given you an example of genetically engineered humans. I've given an example of converting a female mouse to a male mouse. What about making Mighty Mouse? Well, what happens if we take a growth hormone gene and we isolate the growth hormone gene using genetic engineering, and let's say it's from a rat, and we stick it into a mouse, and let's just say we engineer that growth hormone gene so that when you add a metal to the water, let's say you add copper to the water that the mouse eats, the gene gets switched on. We can do that. We can make a gene so that it responds to a chemical. So we take this rat growth hormone gene and we do all this magical genetic engineering in the laboratory, and we say it only can switch on if you put a little copper in water. If there's no copper, nothing happens. So that means you have a genetically engineered mouse, drinks normal water, develops totally normally. You have a genetically engineered mouse that now has a rat growth hormone gene in it. That hormone gene works everywhere in a super way. We've engineered it to be an absolutely super gene. We've done this all in the laboratory by techniques you're going to learn in this class. And now we have that mouse drink that magical water with that little copper in it, and notice we get Mighty Mouse, awfully bigger than the little mouse. So now we've engineered for body size. And imagine what we can do if we started playing around with this with human beings. There's the egg of the mouse. There's the needle. The DNA's in there. The rat growth hormone gene goes in there. We get a big mouse. And we've genetically engineered it, and it's passed on from generation to generation. Well, all that stuff is kind of baby. But now let's start changing the architecture of an organism. Taylor, would you say that the eyes are on the head of an animal?
Yes.
Have you ever seen an animal where the eyes are not on its head?
No.
Well, you're looking at it right there. What do you see in that picture? What do you see, Dora? What do you see, Faria? What do you see in that picture? Where are the eyes on that fly?
The leg.
Oh, did you ever see a fly with eyes on the wouldn't you like to have eyes in the back of your head?
No.
Well, I would. The point here is that now we're getting a little bit more sophisticated. It's easy to engineer a plant to be able to have a protein which the insect bites down and dies. It's easy to engineer a mouse in which when it drinks this magic water it gets big because there's an overactivity of growth hormone because the gene is a super gene. But now let's change the architecture of an animal. Let's make by genetic engineering an animal that doesn't just have eyes on its head, but has it on its leg, or it can be on its wings, or it can be anything else. And furthermore, let's genetically engineer that fly with a human gene. So let's take the gene out of a human being that we know controls the formation of our eyes. When I say controls the formation of our eyes, it's active when we're developing, and that gene is active in the developing head, and that gene is active in the place where our eyes form, and that gene controls other genes that make all kinds of things necessary to make your eye. And we take that gene from a human and we stick it in a fruit fly, and now we engineer the fruit fly and we say, we don't want that eye gene to be active in the head. We engineer it so it's active on the leg. You get an eye. You get an eye on the leg. Rose, do you think those eyes see? Think the eyes on the leg can see like the eyes on the head? Why not?
Because even though there's eye tissue there, that doesn't mean that the, like, I don't know if flies have neural connections.
Yeah,
but
the
like
neural connections are not there. That's exactly true. So you can see the tissue that makes the eye in the organization, but they don't visualize anything because it's not connected to the brain in any way. But maybe we could do that at some particular point. But the point is that you can use genetic engineering to do almost anything. So let me go back to UC Davis. Faria, all of these things that I've said, taking the gene from a jellyfish and putting it in a monkey, taking the gene from a bacteria and putting it in a plant, taking the gene from any creature and putting it in another creature, what does that tell us about all living processes from a genetic point of view? What would you hypothesize?
We have similar DNA and genetic structures.
What would you hypothesize?
We have similar DNA and genetic structures.
Exactly. We have similar DNA. Isn't that what that says, that the structure of DNA in all these creatures has to be the same? Would you agree with that, Faria?
Yes.
And would you say that the processes by which the DNA specifies a protein has to be the same in all of these creatures?
Yes.
Yeah. So what we learned from these genetic engineering experiments is that DNA specifies proteins, and that the process of going from a gene to a protein is universal in all organisms. Do you agree with that, Catherine? Do you agree with that, Juliana? Do you agree with that, Kathy? Yeah, absolutely. That's what that says, experimentally. That's exactly what that says. It says that if you can take a gene from a bacteria and get it to be active in a plant, and a gene from, let's say, a human and get it to be active in a bacteria, it says that all genetic processes in living organisms, from viruses to bacteria to fungi to animals to plants, are the same. They're universal. And that's what we've learned from these genetic engineering experiments, along with millions of other things. So as you can see in these slides, what we've learned is that, as you know, DNA is the substance of the gene, and we'll talk about this more next week. DNA specifies this thing called RNA. RNA provides the blueprint for these things called proteins, which carry out, let's say, the work and makes the hair and makes the skin and makes all the different cells of the body and leads to these things called traits. And we learn that we can intervene in that process. Genetic engineering means we can intervene in this process. And it also means that we can isolate a specific gene, like the glowfish gene. We can stick it in the chromosomes of another creature and it doesn't interfere with anything. The gene works independently, at least this kind of gene. And furthermore, it can replicate. That is, it can divide along with the cells each generation and be passed on from generation to generation. And therefore, we use the basic genetic processes in order to do genetic engineering. Genetic engineering, Davis folks and UCLA folks, is the ultimate organic biology. What I'm conveying to you is that there's no hocus pocus here. We're not inventing anything. We're not making a new process. We're not changing any cellular processes. All we're doing is interceding in the process. It's kind of like getting on an elevator and the elevator just works, except now you're on the elevator that you weren't on before. So genetic engineering just uses DNA processes of replication that we'll talk about DNA process of going from the gene to the protein, and it intercedes in these processes and doesn't change them whatsoever. And that's really important to understand. There's no science fiction. There's no magic. There's no, as I said, hocus pocus. There's no, prayers or anything that you have to say over the cell. You're just using absolutely normal biology, and there's nothing to be afraid of because it's a natural process. The fruit fly with the glow gene in it is perfectly normal. It just grows. It just survives. It lives. It doesn't even know that it has an extra gene. The plant that has the bacterial gene in it that kills the insects, it survives. It grows. It's healthy. It's normal. Nothing happens to it. The girl that has the adenosine deaminase gene that's completely healthy allows her to live. She's normal. She doesn't recognize that she has an extra gene because it's just working as one extra gene. It doesn't turn the cell into a monster. It doesn't turn the organism into a Frankenstein. It doesn't do anything except make one additional protein or two independently of everything else, giving the organism a new trait that it didn't have before. That's what genetic engineering does. It intercedes in this process in a very normal way using these particular rules. Now, before I move on, the next few minutes, what I want to say to you is something very serious. And I want the UC Davis folks to listen to this, and I want all of you guys to listen. And I believe this as passionately as I've lived my 64 and a half years. When it comes to science, there's no magic. When it comes to science, there's no hocus pocus. When it comes to science, there's no mysticism. When it comes to science, there's absolutely no belief. When it comes to science, there's no ether. When it comes to science, there's no opinions. When it comes to science, there's a process. A very rigorous process called the scientific method. There's a process. It's not, I believe. Oh, I think ginkgo biloba. Man, I really take that all the time. It makes me feel really good. Well, there's absolutely no evidence for that. Or, I drink this really great bunch of jamba juice. It's organic, man. It's good for me. Well, what's the evidence for that? And what you're going to learn in this class more than anything else, I don't really care, and John doesn't care. We care, but we don't. I don't care whether you remember the structure of DNA. I don't care whether you remember DNA to RNA to protein. I don't care whether you know about genetic engineering. I don't care about one thing. But what I care about is you come out of this class learning how to think critically. I come out of this class, and when someone comes to you and says, I'm drinking this stuff that's got ginkgo biloba in it, man, I feel like $1 million. What I hope you learn is say, what's the evidence for that? Do you have any data that supports that? Have you carried out a double-blind experiment of people who have taken a placebo and people who have not taken it and shown a difference? That's what I care about. Because not only are you living in an era in which science is booming and moving at an unbelievable speed, but you're also living in an era where people are still perpetuating a lot of anti-scientific thoughts and believe in a lot of New Age nonsense. And believe me, you're not going to hear any political correctness from me for the next 10 weeks, I can assure you of that. The bottom line is that science is a process. You start with observations. From those observations, you construct a hypothesis about how you think the observations came about. You provide an explanation for your observations. Those explanations lead to a prediction. You have observation. The sun shines and comes up every morning. So the sun comes up every morning. You have a prediction. Sun comes up every morning. You get up the next morning and you see if the sun comes up. That's your experiment. So you have a hypothesis from observations that explains your observations. You have a prediction that emanates from your hypothesis, and you have testing that you do experimentally in the laboratory to falsify your hypotheses, to eliminate all the different possibilities. And so you say, what's the question? That is, what's your hypothesis? What's the question? What are your predictions? How do you test those predictions of whether they're correct or incorrect? By working in some way. And then you have new data. And when you get new data, sometimes they lead to new hypotheses. And it goes on and on and on. And that is a scientific method, and that's what this is all about. How do we know that DNA is the genetic material? Well, I just showed you today. We took naked DNA from a jellyfish, naked DNA that produced a protein that when you shine on the protein is green. I took that piece of naked DNA and I stuck it experimentally in a fruit fly, and the fruit fly now shines green. That says that DNA specifies that trait. So that's a scientific process. That's a scientific method. And so the point here is that one needs to be able to carry out this process, and that's why science is so rigorous, and that's why we've had so much advancement in science. And I would venture to say, in all deference from you guys on the North Campus, of which I admire, and all the things that you can do, and all the things that you think about, and all the things that you read, which I frankly can't even begin to understand, science has changed your lives in more ways than anything that has ever been created on the face of this earth because of this process. Because it's not simply, I believe that it's so. It's simply because I hypothesize, I carry out experiments, and then once the experiments are done, they're verified by other people. They're peer-reviewed and repeated until they become established. And of course, as the technology changes, technology is the application of scientific knowledge, of course your observations change, and therefore you have to change your hypothesis. So the point here is that it's not hocus pocus. It's not based on a belief. It's not based on your opinion. And in this class, both here and up north, everything is what's your hypothesis? What is your prediction? How would you test it? Because that's the thinking that I want you to try and learn. Because scientific thinking is valuable in anything you do. Whether you go on to the law, whether you go on to the business, whatever you do, it's valuable. Right, Harry? It's a catharsis, isn't it? Exactly. Okay. Let me shift and let me wrap all this up. Ooh, this one's a little heavier. There you go, Mike. What do you got there, boys? What are the colors? Hold it up.
Cabbage.
What do you have there, Lifen? Hold it up. I can't see it. Hold it high.
Green cabbage.
Green cabbage. What's your hypothesis for the difference in the color between those cabbages? Different genes, or what did you say, Catherine, earlier on? We got to get more microphones. Here we go. Go ahead.
Minor differences.
Minor differences. So, Catherine, what's your hypothesis? Different genes or minor differences?
Minor differences.
So, what you think there's one or more genes specifying this color, Mike?
I'd go with
What's the
more than
simplest hypothesis?
One.
It's always the simplest. It's called Occam's razor. Always the simplest explanation is most likely the correct one. More than one or one?
One.
What happened sometime back, let's say in some field someplace, to the cabbage that was red that's now green? What is your hypothesis, Samantha?
Well, I feel like mutations are
Okay, you feel like it?
Mutations bring about
How do you feel about it? Tell me how you feel about a mutation.
I don't feel about a mutation.
Yeah, I don't either. Thank you.
What
okay?
I've learned. Okay, what I've learned.
No, I don't want you to tell me what you've learned. I don't want you to tell me what you feel. I want you to tell me one thing, what you think. That's what I care about.
There's a mutation?
Exactly. That's exactly the way I want you to say it. There's a mutation. Is there a mutation of the green gene or of the purple gene? What's the simplest explanation?
Green? There's a mutation in the green to get purple.
Ah, you think that's simple. You think you can take something which is green and make it purple, or take something that's purple and make it green?
Something green and make it purple.
What do you think, Anna? Would you agree with that? What's the simplest explanation? Purple to green or green to purple? You think it's harder to make a color? Green's not really a color. You think it's harder? Can I have this thing? You think it's harder to make this thing?
Yes.
Harder.
Yes.
Do you agree? Where's your little nameplate? What's your name? Derek, get your name up there. You think it's harder to make the purple or the green? What do you think, simplistically? Dora, what do you think? Faria, what do you think? Harder or easier? You think that the plant has to work harder to make it purple than make it green? What do you think? What's the color of most plants?
Green.
Yeah, you think it's harder to make it purple or easier? I know
Harder.
I'm using an anthropomorphic term, but what do you think?
Harder.
Yeah, how many would say harder? Exactly. So what do you think the mutation was in going from purple to green cabbage? You think the purple gene was mutated or the green gene was mutated? What?
Green.
How many say purple? How many say green? How many say purple? How many say green? How many don't know? It's about even. It's actually the purple gene. So imagine that you had a mutation. Do you think, Elaine, no, I've called on you, Maura, do you think that the mutation would occur just spontaneously someplace?
No, wouldn't it be?
I don't think that's on. You think that the mutations would occur spontaneously? Could you make sure they're on before you give it to them? Go ahead.
I don't think it would occur spontaneously.
You don't?
No.
Can you look around the room? Could you stand up, please? Oh, God. Can you stand up, please? Can you look around the room? Do you look the same as everybody else? Why do you think you look different than everybody else in the room? Do you think you have any different genes than Elizabeth's genes? Do you think you have any different genes than Michelle's genes?
I think no.
Yeah. Why do you look different?
Different arrangement.
Well, let's say different chemical forms of the same gene.
Yes.
Do you think that all of this diversity that we see in this room, this genetic diversity, arose spontaneously, or someone created it?
Oh, okay. In that sense, spontaneously.
Yeah. So do you think that going from this purple thing to this green thing was spontaneous?
Yes.
Yeah. So what do you think about, you can hold that down, what do you think about the fact that genetic engineering is nothing new? What do you think about going to the grocery store, whether it's Ralph's, Vaughn's, God forbid Whole Foods, and looking at all those vegetables? Do you think any of those existed in the wild? The tomatoes that you eat have never existed in the wild like that. The broccoli that you eat never existed in the wild. The potatoes that you eat never existed in that form in the wild. Every single solitary vegetable that you eat was engineered way back in time. Do you think that a cat existed in the wild way back when? No. You think a dog existed in the wild? You think all those breeds of dog, Great Danes and Chihuahuas, and these little yapping, little ratty things, you think all those dogs, they were just out there in the woods, yapping at those horses and everything else that were out there? How do you think that those things were created? Yeah, they were created by genetic engineering. Maybe it wasn't as sophisticated as the genetic engineering that we do today, but what do you think the breeders did when they bred for a Chihuahua compared to a Great Dane? What is your hypothesis, Jessica? What is your hypothesis? What were these breeders doing when they were selecting for the Chihuahua versus the Great Dane? What were they selecting for? What's your hypothesis?
They looked at the traits they wanted.
And does that mean they were looking at the genes they wanted even though they didn't have them in their hand? What's a trait determined by? What's a trait determined by?
The parents?
What's a trait determined by?
Your DNA.
Yeah. What's the trait determined by? Ultimately, the gene. So do you think when they were selecting for this Great Dane and this little yapping, yap, 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 you know those dogs, I hate those dogs. Can't stand them. Keep you up all night, this yapping constantly. What do you think they were selecting for, Jessica? Give me a hypothesis. Put the microphone up and give me a hypothesis. Simple. You just said it.
They were selecting for the genes they wanted?
They were selecting for the genes they wanted. They didn't know what those genes were, you know, thousands of years ago, but they were selecting for the traits. And it just so happens that these dogs come from wolves. And so at some point, 10,000 years ago, someone said, I got a wolf, and maybe they recognized that it was a little tamer than the other wolves, and they brought it into their little place that they were living and started breeding it, and that trait was then perpetuated. So the point here is all farm animals have been domesticated. All pets have been domesticated. Every single solitary vegetable you eat in the store has been domesticated and has been, Hannah, can you get that? What is that? Can you hold it up? What is that? Eric, what is that? What is it?
Corn.
Yeah, it's corn. You think that that stuff existed in nature? Seriously. You think you can go in the woods and see, oh, here's this corn, you know, this is really great. I'm going to buy it. This is going to be fabulous.
No.
Yeah. This stuff does not exist in nature. I'm serious. This stuff was engineered. What are these yellow things, Elizabeth? But what does the kernel do?
I guess they're the seeds.
They're the seeds. You think the seeds should fall off of this thing?
Yes.
But they don't. How is this thing going to survive in nature? Is this a fruit or not? What is this cob? It's the fruit.
Right.
The seeds develop in the fruit. These seeds never fall off the fruit. How is the plant going to perpetuate itself? It can't. This thing has been engineered from a wild variety of corn that has a cob that's this big and has 3 seeds on it. It's been engineered by breeding, by selecting traits. We now know what the genes are. Can you guys just move to the door, please? Yeah, thank you. I don't need an audience up in front. Thank you very much. I like audiences, but in front of me, not behind me. Okay. The point here is that everything, there you go, Mac. Whoop. Woke you up, didn't I? The point here is that we've bred everything. Now, I want to say something. I want to make it clear. When our ancestors bred for all of these different things, they knew what they were doing. Just because someone lived 10,000 years ago didn't mean that they were bright. They were. They just didn't have the scientific knowledge to know the basis of what they were doing, but they had the insight and they had the creativity to know that they wanted to survive. And in order to survive, they wanted more food. And in order to get more food, they wanted to engineer the plants to be able to give more seeds, like the corn. There's the ancestor corn. How would you like to be growing that? You think you get more seeds out of that? No. The point is that they were engineering traits. They were manipulating genes. And they were doing it by classical breeding. Whether it was plants, whether it was animals, notice that we have on the screen a bunch of different tomato fruits. I want you to all switch on and look at this, both up in Davis and here in Los Angeles. This is one of the most important things that you're going to learn this entire quarter. What you see is different shapes, different colors, and different size. That's called genetic variability. I want you to all look around this room. Seriously, look at your neighbors. Look at your fellow students. We look different because of genetic variability. Those tomatoes have the same genes in their genomes. There's no differences. We have all the same genes, except for males and females. But yet we look different. And we look different because of this thing called genetic variability. Genetic variability. And what genetic variability is in its simplistic form, genetic variability is due to random mutation. We take a gene A and we mutate it to gene A1, we mutate that to gene A2, we mutate that to gene A3, etc. It's the same gene, just a slightly different form. And that slightly different form makes brown hair, blonde hair, brown eyes, blue eyes, hazel eyes. Same gene, just slightly different form that occurred spontaneously in nature. It's like, for example, how come all of us in this room, well, most of us, I won't say all of us, but most of us, let's say, have fair skin. I won't call it white skin because that's not really politically correct. But we have fair skin. There's no African Americans in this room that I can see. There's no dark skin-colored individuals. Well, how come all the individuals, most of which in, let's say, Africa and parts of the world, have dark skin, but the ones that live in Norway and Sweden have, where did that come from, Michael? Where did that come from?
The environment that they lived in.
Well, how did the genes specifying darkness turn into the genes that didn't specify darkness? Any different than purple and green? What's your hypothesis?
I don't know.
Where did we originate from? Do you know? All human beings.
Yeah.
I can't hear you.
Mesopotamia.
I don't know. I don't think so. I don't even know where Mesopotamia is. What I want you to do is deconstruct all these things you've memorized for these multiple choice tests. And what I want you to do is start thinking. Well, we all originated from Africa 200,000 years ago. And I can assure you that our ancestors were all very dark-skinned. Well, how did all these people in the world get to have white skin? Was it a mutation? What do you think? What's your hypothesis? Kathy? You think? What's your hypothesis?
My hypothesis
Thank you.
is that as the environment where they lived shifted, their genes had to...
You think the genes responded to the environment, or you think that all of this variability just happened spontaneously? What do you think, Nancy?
I think that
You think that all of this variability in the structure of these genes just occurred? Or do you think someone ordained them?
I think initially there was just a spontaneous
Yeah,
mutation.
say it again and louder. Thank you.
Initially there was just a spontaneous
Yeah, initially they were spontaneous. And then, because of maybe the environment, it just so happened that, you know, you need vitamin D in order to survive. And, you know, if your skin is really dark and you live in really northern parts of the earth, and you don't get enough sunlight, particularly in the winter, you're going to get some serious growth developmental defects. And so there was a selective advantage to be able to have skin that didn't reflect light and be able to make more vitamin D. The point here is, and again, as I said, this is one of the most important things you're going to learn this entire quarter, is that just like us, these tomatoes, all of this variability occurred spontaneously. Chemical changes in the same gene. Chemical changes, mutations in a gene that alters slightly the function. So instead of having a red tomato, you have a yellow tomato. Instead of having a round tomato, you have an oblong tomato. Instead of having blue eyes, you have brown eyes. Instead of having blonde hair, you have brown hair. Instead of having dark skin, you have light skin. Same genes, slightly different form. So what our ancestors did in making the horses and the goats and the chihuahuas and all the other vegetables, where's the broccoli? Okay, where's the cauliflower? What is this? There you go, Elizabeth. What's that? Hold it up.
Kale.
Yeah, it's kale. Do you think kale is related to this? Does it look the same? Looks totally different, doesn't it? Where's the broccoli? Eric, does that broccoli look like this? Does the kale look like this? Audrey, are they the same plant or a different plant?
They're different plants.
They're different plants, totally different, like monkeys and humans.
No, they have the same.
Say it louder.
They probably have the same gene. With
They
slight
probably, or they
mutations.
do,
They
or
have the same gene. With
is it the
slight
same
mutations.
species, or is it different species? What's your hypothesis?
It's the same species.
Exactly. These plants are exactly the same, but they look different. And they look different because that's the one that looks like what was in the wild. Do you want to eat that thing? Well, you can, but you'd be eating the leaves. When you eat this, you eat the flowers, and there's a lot of nutrition in the flowers. And so what our ancestors did was to breed these plants to have more flowers, more food, more nutrition, and survival of the species. And so the point here is that when our ancestors genetically engineered animals and plants, what they did was they looked for different traits that were specified by genes. And now I'm going to introduce a new word, an esoteric word, which is one of the most important words that you need to remember the entire quarter, and that's a word called allele. Very fancy word. What it means is different form of the same gene. And we're going to talk about this over and over and over and over and over again. And it's different alleles which allow for genetic variability. That's what creates genetic variability. Alleles arise, different forms of the same gene, A, A1, A2, A3, A4, A5, A6, all occur by spontaneous mutation. But they pre-exist in the environment. And so all of these different tomato varieties, or all of these different brassica varieties, can be selected because these mutations occur pre-existing. So more than that, let me wrap all of this up. The early bioengineers took a tiny tomato that if you bit down on this tomato, you would spit it out in about 2 seconds because it's noxious, has a lot of toxic stuff. The reason why the wild tomato has a lot of toxic stuff is it fights off insects. But the breeders bred it to be larger because of the fact, Daisy, can you just calm down, please? Breeders bred it to be larger, more food, bred away the noxious chemicals, and made it tasty so one would want to eat it. And it's all because of existing variability. Same with the corn. Here's the ancestor, few little seeds. Here's the one that we've domesticated, lots of seeds, and so on and so forth. There are the different brassicas, all selected for. Same with animals. Now, let me just wrap this up. Classical breeding is manipulating genes. The problem with classical breeding, and I'll be done in a sec, the problem with classical breeding is you can't predict the results. And so a classical example of this is, and I like to tell this story a lot, is that one of the first plant genetic engineers decided that he was going to take the cabbage and he was going to take, where's the cabbage? He's going to take the cabbage, he's going to take the radish, he's going to cross them together to get a plant that has the head of the cabbage and the roots of the radish. That's a very valuable plant because now you increase the food supply twofold. Because on one plant you can eat the root and you can eat the head. And this guy by the name of Karpachenko lived about the time of starvation in the new Soviet Union, and he was engineering the new plant. The problem is when he did that, notice what he got. He got a plant that had the head of the radish and the roots of the cabbage. Virtually useless. It's a new plant that he engineered by using breeding, but with no predictable results. Now, remember the results that I showed you with the glowfish. Took that one particular protein, put it in a Drosophila, shined a light on it, and I got one thing happening. Very predictable, very, very accurate, very specific. The point here is that genetic engineering, either done the classical way by breeding, or being done the molecular way, both manipulate genes. The difference is, with the modern way of genetic engineering, we know what the gene is, we've isolated the gene, we understand what it does, and we can get very specific and accurate results, almost with no ifs, ands, or buts. The old-fashioned way is hit and miss. It's still very important. We still use it. It's still critical, but it's cumbersome. It takes time, and there's one important thing. The old-fashioned way, you only can engineer tomatoes with tomatoes, dogs with dogs, tomato plants with tomato plants, as I said. With genetic engineering, every single thing is possible, as I showed you. There's no limits. None. It's accurate, it's safe, it's effective, it's exciting, and it's just a tool. And this is what I want to end with. Genetic engineering, whether done by breeding, Dora, Faria, either done by breeding the classical way or being done by modern techniques, is just a technique interceding in natural processes. The purple cabbage gene exists, and we know what it is. We can turn a green cabbage into a purple cabbage by using modern genetic engineering. We're no longer manipulating genes by classical breeding. We're manipulating them in a very directed way. Genetic engineering is directed change. And that's what we're going to talk about for the next 10 weeks, how it happens, why it happens, ethical aspects of it, legal aspects, and everything else.